My name is Andrea Hernandez. I'm a biology major here at Florida Atlantic University. For the past 3 years, I have been conducting research at the Florida Atlantic Biomechanics Lab. With this research in biomechanics and functional morphology, I have really focused on aquatic locomotion in sharks specifically. With these research, we look at the ecology and evolutionary traits that have helped shape these forms of locomotion so that we can better understand these species. My career goals are to potentially continue my master's here at Florida Atlantic University, earn my master's degree, and continue research in marine animal conservation using my knowledge of functional morphology, evolution, and ecology. As part of the Summer Undergraduate Research Fellowship for the summer of 2017, I conducted a project on the ontogeny of morphology and muscle development in 2 benthic elasmobranch species. So looking into elasmobranch locomotion, we're looking at the way that sharks and rays move underwater. One of the really big qualities that we look at when we look at locomotion in sharks and rays is their fin morphology. Their fin morphology really helps stabilize and create different forms of locomotion in these species. In benthic species specifically, they have to have more flexible fins. So what that means is that they have more reduced skeletal elements in their fin, and they're more spread apart, as well as different forms of muscle and different muscle attachments than would more pelagic species. So they have really flexible fins to navigate on sand substrate, reef substrates, or rock substrates. The 2 species that we looked at in this project were the epaulette shark and the bamboo shark. These 2 species are really closely related. They're the 2 genera of this one family, Hemicillidae, and both these species live in shallow benthic environments in the Indian Ocean. The epaulette shark is really restricted to shallow reefs, so they have to navigate these really tight-knit reefs to find their food that is often buried in the sand or within crevices. Whereas the bamboo shark is a little bit more spread out, and they sort of navigate reef environments as well as sand and substrate environments. So the 2 sharks that we are looking at have 2 forms of locomotion. They can swim, and additionally, a lot of observations have been made on them actually walking out when they come out of their egg casings. And so they walk on these 4 fins that they have, their pectoral and their pelvic fins. But what's not really well understood is how similar or different these 2 forms of locomotion are in these two species, and also if the morphology differs with these different forms of locomotion in these 2 species. So to get a better understanding of this, we looked at 2 objectives for this project. The first is to understand muscle movement and development during the embryonic stages of these 2 species. We were looking at how the muscles develop in these egg casings of the 2 species as their embryos, so that we can understand how that muscle changes and moves and how much they can move in their embryonic stage compared to what we already know that can happen in their juvenile or hatchling states. The second objective was to look at how the morphology, the actual anatomy of the muscles and skeletons differ through embryonic stages between these 2 species, so that we can get a better understanding of how the ecology of these habitats have shaped the morphology in the fins of these 2 species. And so we conducted 2 types of experiments to do this, or we used 2 different methodologies to look at these 2 different objectives. For the muscle movement and fin movements, we conducted muscle stimulation experiments so that we can stimulate the muscles of these embryonic sharks, so that we can see how much they move at different stages of their embryonic development. And then we can calculate how much they rotate by measuring the distance that these fins move while they were being stimulated. So we can calculate the different angles of rotation and different forms of movement that they did with these fins when we stimulated those muscles. Another really cool technique that we used was clearing and staining. And so what we do with these specimens is that we clear sort of the viscera and integument so that you can see through the animal, and stain the cartilage in their skeletal system so that you can visualize the fin morphology within the shark. So that gives us a really better, clearer picture of how the morphology looks within these fins. And so from our muscle experiments, what we found was that we can actually successfully detect these muscles and stimulate it. Unfortunately, we did not have all the specimens that we needed to compare these different muscle stimulations in different species, but we did have one bamboo shark that we were able to obtain, and we stimulated its muscles, and we found that we can accurately find the muscle, stimulate the muscle, and calculate its range of motion. And so what you see in this poster here is the first graph is sort of the different points on the fin that we looked at and how they move, and sort of how they moved in relation to the different points on the fin. And then also the second graph shows that when we stimulated the depressor muscle, the fin moved down as we would expect. So that's really encouraging results that we can actually stimulate it, so that in the future we can continue these muscle stimulations and hopefully compare it within the embryonic stages of a species and then between these two species to get an idea of how much these muscles development and how they move differently and how they develop these different muscles. With our clearing and staining methods, we were able to clear and stain the fins of these two sharks species, and so that's what you see here in these pictures. The first picture is the pectoral fins, and so you'll see the bamboo and the epaulet shark next to each other. And what we found was that from one bamboo embryo and one epaulet juvenile that we were able to obtain, that the epaulet shark actually has a much more flexible fin morphology in both the pectoral and pelvic fins. And so in the pectoral fins, they have what are called basal cartilages, and so those are the bigger, longer structures, more proximal, so more attached to their body midline. They have just two basal cartilages with this gap in between that allows for more muscle attachment and also potentially more gripping of these rock and coral reef substrates that they have to navigate. Whereas the bamboo shark has three basal cartilages that wouldn't allow as much movement and flexibility to grasp any rocks, but they don't have to because they live in sort of substrate and sandy environments that they can navigate, so they don't have to be as exclusive to those coral and rock habitats. Additionally, the end segments, so the radial segments, are also more spread apart in the epaulet shark, and so that they can also navigate the reef better. They have more space in between, so it would be the difference essentially between the long bones in your hand and the shorter bones in your finger versus the long bones in your feet that make your foot a little bit more stiffer and the little tiny muscles in your toes. So if you think about the bamboo having sort of more foot-like structures where it's a little bit more stiff and maybe not as flexible, and the epaulet sharks have more flexible fins like a hand so that they could grasp the rocks better and get around their environment. And so some of the future work that we hope to do with this project is to get a larger sample size so that we can actually compare the muscle stimulation experiments between the different species at different embryonic stages, and additionally clear and stain a few more specimens to see if we keep seeing the same pattern in morphology differences, if they do really in fact have more flexible fins in the epaulet shark versus the bamboo shark. And of course, I always have to thank people because it would not be possible without all the support. I would definitely like to thank the Summer Undergraduate Research Fellowship for funding this project and all the moral support and incredible mentoring that they provide, as well as the Florida Atlantic Biomechanic Labs and all the graduate students and Dr. Marianne Porter for always supporting us and coming up with these really creative and different scientific questions that we come up with.